Thanks very much. It's my pleasure to be here. Thanks for the invitation and for humoring a stereotactic and functional neurosurgeon from speaking at this illustrious meeting. So thank you. I joined last year virtually. I wasn't able to make it, so it's my pleasure to be here with you this afternoon. So my name is Lipsman. I'm a neurosurgeon at the University of Toronto and a functional neurosurgeon by training. And over the last 10 years, we've been working on focused ultrasound and MR-guided focused ultrasound, which you have heard a little bit about in the earlier talks late this morning. And I'll talk about some of the work that we've been doing in this area over the last decade or so, where we are now, where we're heading in the next few years. So I often start by showing this first page of a paper from the mid-1950s. It was the first time that focused ultrasound or ultrasonic irradiation was used for lobotomy. Of course, we don't use that term anymore. It's frowned upon in the neurosurgical world. But suffice it to say, this is a very sort of established and old discussion that we've had in neurosurgery for a long, long time. So a few things. I mean, I could spend a long time talking about this paper, but a few things that I'll highlight. The first is that 70+ year history of trying to use ultrasound therapeutically in neurosurgery. So a very rich tradition, which I'll go into in a little bit. So this is a problem that's been tackled for a long time. There's always been a need for this. So at the time, it was treatment-resistant mental illness, neurologic conditions. So the clinical need has always been there for a safe and effective way to intervene in subcortical areas for influencing disease circuitry. At the time, though, in the 50s and afterwards as well, the bone represented a major obstacle, a major issue for ultrasound. So, of course, the bone will attenuate 99.9% of ultrasound energy. Any energy that passes through the bone is highly diffracted, very difficult to then refocus onto discrete parts. So a craniectomy was required to really, enact any kind of lesion at that time. At the time, you can see schematically a single transducer was used, and now we're, as you'll see, we're at least 1,000 transducers. And ultimately, this was the goal shown in the 1950s, and I'll show you some imaging from today. It's a discrete lesion that preserves areas around that lesion in a way that can be very focal and safe and effective. So there is, again, a rich history of focused ultrasound. The first time it was used really in the literature, the term was in the 1940s by John Lynn, who is a neurologist at Columbia. And we've evolved significantly from single transducers to multiple transducers and ultimately with pairing it with MR-guided focused ultrasound to over 1,000. And what this has resulted in is, very much what this specific meeting that we're at is trying to get at, where minimally invasive and subcortical surgery tries to get to, which is really a disruptive technology. How do we replace tools that are currently being used or create potentially entirely new markets where they don't exist? So that's really where we want to be as a field and as practitioners and as scientists and as clinicians in this area. And focused ultrasound, as you'll see, really has that potential for disruption. So when we looked, a couple of years ago at the field of focused ultrasound, specifically in the brain, so these are intracranial applications of therapeutic ultrasound, and we mapped the number of publications per year specifically in that space, and that's that red line on the graph. You can see that there's been an exponential rise in academic interest in this area. And the inflection point was really in the early 2000s, where a lot of the initial work was being done with the first mechanisms of using ultrasound in things like chronic pain and things like tremor, as you'll see. So a lot of the advances have been driven by the clinical brain application. So we've used ultrasound for other parts of the body, but a lot of the clinical advances currently are being driven by the intracranial applications. So when we talk about ultrasound, acoustic energy at frequencies much higher than we can hear, of course, the two most common mechanisms, and the one that I'll touch on in this talk, are high frequency, also known as high Fu, and low frequency, also known as low Fu. So high frequency ultrasound generates a discrete lesion on the brain, very similar to LIT or to RF lesions, histopathologically and otherwise. It's really radiocoagulative necrosis that we cause with a high frequency lesion at high amplitudes, high powers. With low frequency, we're using much lower energies, much lower frequencies. And here, we're aiming for a transient permeabilization of the blood-brain barrier. Okay, so we're doing it in a very focused way. And there have now been at least 300 or 400 papers looking at small to large animal models that have shown that, in fact, you can use ultrasound, low Fu, to generate temporary safe openings in the blood-brain barrier and deliver myriad different therapeutics to the brain from antibodies, vectors, cells, and chemotherapy. Now, there are other mechanisms. I won't touch on these in too much detail, but there are at least 14 or 15 different mechanisms by which ultrasound energy can interact with the brain. There are neuromodulatory indications, histotripsy, which is very finely cut lesioning strategies, targeted uncaging of nanodroplets that you can load with chemotherapy, as well as radiosensitization and hyperthermia that you use together with radiation therapy to have an impact on the angioarchitecture of things like tumors and other neurodegenerative conditions. So all of these at various stages of development and various stages of going into the clinical space. So three things that I'll touch on over the next few minutes. The first is the expanding indications in the field of focused ultrasound. Where are we now? Where are we headed? Look at therapeutic delivery, which again, Dr. McDermott and others have talked about in terms of delivering chemotherapy and other things to the brain, and how can we personalize these approaches as much as possible. So when we talk about MR-guided focused ultrasound, of course, body uses. So everything outside of the head, body uses have been very well established for the last 20 or 30 years. So we use ultrasound for diseases of the prostate, of the breast, of bone metastases, fibroids, et cetera. But only relatively recently has the bone problem been overcome. And this has been left to much smarter people than me, the physicists, et cetera, who are able to overcome this idea of bone attenuation and how do you refocus that energy when you get it through the skull. So we know that there's a lot of diffraction and visualization that is required as well. So what we needed as a field was a central millimeter-sized target with a very clear behavioral assay that we can then test these indications for. And that made essential tremor really the optimal application. So what are the advantages of focused ultrasound lesioning in the brain? The first is that it's real-time MR thermometry, that we can actually see a lesion develop before our eye. We get feedback every 3 seconds about how hot the tissue is getting. We can see the lesion develop in the MRI scanner within seconds, which gives it an advantage over something like gamma knife radiosurgery, which takes a few weeks or months to develop. And as again, we do the procedures awake, so there's a real-time behavioral assay. We can test the patient during the procedure and see if we're getting adverse effects or if we're getting any clinical responses. So maybe if we can start the video on the left, in the back. So essentially, this is a gentleman recently treated with a very typical essential tremor affecting his right upper extremity, typical postural and appendicular tremor, which affects most of these patients in the dominant side, highly treatment resistant. In a procedure that took approximately an hour and a half to 2 hours, we generated a lesion in the left-sided VIM nucleus. And yeah, if you want to play that video on the right, essentially after the procedure, still in his gown, the tremor is completely abolished, and patients essentially go home within about an hour and a half or 2 hours of the procedure. Okay, so this procedure has now become ambulatory. So together with our colleagues at UVA and at 8 centers around the world, we conducted our RCT looking at ultrasound in tremor a few years ago that found really without a shadow of a doubt that this is an effective treatment that is very comparable to other surgical strategies, including DBS and RF thalamotomy. It's not without its side effects, of course. Real estate is everything in the brain, and where we make these lesions is important. When we look at the side effects, the most common ones are going to be sensory because of the proximity to the sensory nucleus, and it's going to be a gait associated with sort of largely cerebellar proprioceptive issues related to feedback from the deep cerebellar nuclei to M1 motor cortex. Importantly, though, the side effects are very similar to what we see with DBS and with radiofrequency, so it does not appear to be any riskier. So just to give you a sense of how disruptive this has been in our practice, so we asked the Research Ethics Board at our institution to conduct the first trial, which was really only the second site anywhere in the world other than UVA in 2011. We treated our first patient 6 months later in 2012. We published our first report in tandem with the UVA group in 2013. Took us a few years to do the RCT. In 2016, we got regulatory approval, both at Health Canada and FDA. So that period of time, that's 6 years roughly, or about 7 to 8 years from initial idea to commercialization, is about a third of the time it takes a typical drug or device to get to market. Okay, so that shows you the disruptive potential of this in our practice. So we've now treated nearly 500 patients at our institution. This is fully ministry funded, Health Canada approved, and we've gone on to conduct staged bilateral treatments which in the world of functional neurosurgery was really anathema for ever. You know, this idea of making bilateral thalamic lesions was not done, but now we're doing it in a staged fashion because we can precisely control the size of the lesion that we're making. So this got us thinking, you know, there are other conditions for which making a lesion may be effective and safe. And of course, the psychiatric conditions are those as well. There's a very robust and long history of using precise lesions for treating patients with OCD and depression, specifically capsulotomy and cingulotomy. So again, decades-long history going back to the 70s and 80s of using radiofrequency probes in order to generate lesions in the capsule for mood and anxiety disorders. But typically the lesions look like this with RF, okay? Very large and up to 30% rates of neuropsychologic, cognitive, affective, and speech side effects. So led to a huge backlash in the psychiatric community about actually using this for patients who would otherwise benefit. But now we can make lesions that look like this, you know, very discreet, minimal edema, image guided, and quite precise. So we can really target the anterior limb of the internal capsule in a way that is really quite safe and effective. And now we've shown this. This is work that was done by Dr. Davidson, who is doing a PhD in my lab, and Dr. Hamani at Sunnybrook. And we've shown that in patients who are otherwise really refractory, truly treatment resistant, we see significant improvements in anxiety scores over a period of a year, such that patients who are on average ill for about 20 years by the time they see us are treatment responders in the majority of the time, about 50 to 60% of the time by 12 months. So it really has become a salvage procedure for these patients with no real other treatment alternatives. So what is the result of that? Well, we've really used this as an opportunity to spread the word about psychiatric neurosurgery, specifically in this context. We've established a multidisciplinary clinic that's become a provincial and national resource in Canada. We treat patients coast to coast, and we see this as really the tip of the spear. So not everybody gets focused ultrasound that's referred to us, but it gets them in the door. It gets those patients that are highly refractory, very, very sick, with a very stigmatized illness. At least they get in the door, and whether it's focused ultrasound, whether it's TMS, ECT, DBS, at least they're getting some kind of treatment. But this is a way to sort of ensure that we validate their illness and their need for that. The procedure is not without its limitations, of course. Not everybody's skull, we quickly discovered, is made equal, and the skull density actually matters a lot for focused ultrasound. We know that because the ratio of cortical to cancellous bone will actually influence how effective and efficient ultrasound is able to travel through the brain. Patients with a very low skull density ratio, it becomes very difficult to generate lesions. We also can preferentially make lesions in the geometric center of the brain, and bilateral treatments become more challenging. So all of these are technical limitations for a technology that's very much expanding and growing, and I anticipate over the next few years, each of these will be checked off and be addressed. So a quick word about therapeutic delivery and then personalized approaches. One of the most promising advances in FUS is this idea that we can temporarily open the blood-brain barrier to deliver therapeutics. Of course, the BBB is a major challenge for the delivery of any kind of therapy to the brain. We've seen it in glioblastoma and other oncologic applications, but it's also the case for other diseases, including the neurodegenerative diseases. Many different approaches have been tried. They've all been met, unfortunately, with either off-target effects, either they're very expensive or they're risky. So a way to transiently do this in a way that's safe and effective really has been sought for several decades. And this is how focused ultrasound tries to do this. So we pair the delivery of ultrasound energy with the delivery of microbubbles to the brain. So microbubbles are small gas-filled spheres circulating throughout the body. Once they get to the brain, we expose the region of the brain to focused ultrasound energy. The microbubbles will absorb the energy, and they'll oscillate about 500,000 times a second. So very, very quickly, and they'll physically pull apart the cells making up the blood-brain barrier so that whatever is co-circulating has increased access to pathology where you want it to reach. Okay, so this is a transient process that lasts less than 24 hours. So the first preclinical data suggested that in a mouse model of breast cancer metastases to the brain, that ultrasound can actually be used to deliver Herceptin. Herceptin is a monoclonal antibody that weighs 150,000 daltons, so 150 kilodaltons. The typical drug that can get across the BBB is 400 daltons. So this is several thousand times larger. And in fact, when you looked at the contrast enhancement of these mice that underwent focused ultrasound sonication, the degree of Herceptin concentration varied directly with the intensity of contrast, suggesting that contrast enhancement can be used as a proxy measure for BBB opening. Okay, so this was the first time that we can actually show in a preclinical model that this may have some legs. So what we did, we leveraged that preclinical data and conducted a phase 1 trial looking at Herceptin delivery in patients with otherwise progressive intracranial metastases from HER2-positive breast cancer. So this was a first of its kind trial spearheaded by another PhD student and neurosurgery resident at the time, currently staff Ying Meng. And what we did, we recruited 6 patients. Here you can see on the left one of our patients with a otherwise irresectable tumor in the cerebellar peduncle. You can see in the second panel, the focused ultrasound BBB opening as shown by a kind of color map, increased fluffy contrast enhancement in the area of the tumor, which goes away 24 hours after the procedure. So very focal, close to the ependymal lining, opening of the blood-brain barrier in a way that increases contrast enhancement for less than 24 hours. But what we did in this study is we actually tagged trastuzumab with 111 indium so that we can directly visualize it in the brain. So with a SPECT scan, we can actually see trastuzumab in the brain. So that very same patient here on the left, injected with 111 labeled trastuzumab immediately before focused ultrasound, and you can see that it accumulates in the cavernous sinuses and the venous sinuses, and immediately after focused ultrasound, it's accumulating within the tumor. And we can see about a 450% increase in Herceptin concentration. So this was the first visual confirmation of direct, targeted, enhanced delivery of a monoclonal antibody into the brain that otherwise cannot get in a very focal way. So again, leveraging this, we have very strong interest to work with our pediatric colleagues, and this is work done together with Dr. Rutka at Sick Kids in Toronto, who helped pioneer work looking at doxorubicin delivery in patients with DIPG, pontine glioma, uniformly fatal, of course, in children aged 5 to 18. So we designed a phase 1 trial currently ongoing, looking at enhancing BBB opening in patients with otherwise progressive DIPG in a kind of dose escalation pattern. The dose, though, is not the dose of the drug, it's the dose of the BBB that we're opening, okay? So here's the first patient that was treated in the world. On the left, you see a baseline MR showing a fat pons appearance, typical of DIPG with a biopsy tract on the right. On the second panel, you see that we opened the BBB only in 50% of the pons. So you can see contrast enhancement only on half of the pons, okay? That's the level of precision. 75% of the pons about a month later, and 100% of the pons a month after that. And a typical postoperative MRI scan showing gadolinium enhancement has disappeared within a day. So just to give you a sense of the level of precision, when we open the blood-brain barrier, we open it in axial slices. So this is an example of the treatment console, and you can see multiple axial slices on the left. And on the right, you can actually make out the individual axial slices of contrast enhancement, indicating complete coverage of the pons in this patient. So we can achieve really sub-millimeter accuracy with focused ultrasound and enhancing drug delivery. A quick word about diseases that neurosurgeons don't typically treat currently, but will in the future. One is Alzheimer's disease, of course, another disease where enhancing therapeutics to the brain will be essential. In a first of its kind trial, we opened the blood-brain barrier in a relatively ineloquent region of the brain in the right frontal lobe, showing that in fact in patients who are amyloid positive, we can open the blood-brain barrier as shown by a rectangular area of discrete enhancement that disappears the next day. So of course, all of these vessels are heavily amyloid laden, and we were able to safely open the BBB in this region of the brain. In a subsequent study, there's a video there, but it's not essential. But essentially what we were able to show is that in patients who have amyloid positivity, as evidenced by amyloid PET scans, we're able to open the blood-brain barrier in several regions that are highly eloquent for memory and default mole structures. This was the first patient that was treated bilaterally with bilateral hippocampus, cingulate, precuneus, and parietal areas. So the implications here for the delivery of therapeutics to anywhere in the brain is quite significant, including bilateral hippocampal amygdala, entorhinal and amygdala areas for conditions where you want to deliver any therapeutic to. Another of those indications is Parkinson's disease, and I'll just show this real quick because this is one of the areas where, again, a disease-modifying treatment has been sought for many, many years and is currently not available. So working with our neurology colleagues, we designed a trial where we want to open the blood-brain barrier in the putamen while we deliver enzyme replacement therapy. And the enzyme that we're delivering is called GCase, glucocerebrosidase, which we know is implicated in alpha-synuclein pathology in patients that develop ultimately both idiopathic Parkinson's, but also a genetic form of Parkinson's known as GBAPD. Getting that enzyme to the place where it's needed most is a significant challenge in patients with Parkinson's disease. So here we've shown that we can very precisely, with really sub-millimeter precision, open the blood-brain barrier, in this case in unilateral putamen on the right side. And you can see quite precise contrast enhancement there on the right. And what's interesting here is when we do FDG PET scans a month after the procedure, so again, the BBB is only open for a day, but when we do FDG PET scans a month after the procedure, we see significant reversal of glucose hypermetabolism only in the putamen that was treated. Okay, so there may be some longer-term biologic effect that we're picking up there that may be linked to a disease-specific marker. We're now progressing this to a phase 1/2 trial where the goal is opening bilateral putamen. This is a patient that was treated just a few weeks ago, showing bilateral putaminal opening of the BBB, showing bilateral contrast enhancement in the absence of significant bleeding or swelling. So in terms of personalized approaches, liquid biopsy approaches were mentioned earlier. Why is this important? It's for really lesions like this where the differential can be quite broad from a very challenging surgically tumor that could be malignant to a highly treatable tumor that could be responsive to chemotherapy. So can ultrasound potentially be used to detect peripheral biomarkers of tumors, obviating potentially the need for surgery? The answer appears to be preliminarily yes. So using some of the data from our own trials, we were able to detect at least a 3- to 4-fold increase in cell-free DNA immediately after we opened the blood-brain barrier. And pooling together all of our patients from Alzheimer's, GBM, and other indications, we're starting to parse out different disease states based on methylation profiling of the cell-free DNA that we're seeing. So leveraging this, we were conducting a trial with 20 other sites across North America, including Dr. McDermott's group as well, I mentioned earlier, where the goal is ultimately going to be to enrich plasma for cell-free DNA to see if we can use ultrasound as a potential liquid biopsy strategy. So what are some lessons learned? We've treated about at least 550 times with focused ultrasound for multiple indications. Appears to be a well-tolerated procedure that can be done on the same day with no serious adverse events. Very large multi-target volumes can be treated, including an eloquent cortex. We believe strongly in a kind of progress and parallel approach. You've seen multiple indications here, so we don't wait for one to be done. We're trying multiple indications, multiple therapeutics at the same time. As a result, we invest very heavily in strategic partnerships with industry, with our hospital partners, our research institute. I would say that 90% of the work for all of these trials is strongly regulatory. Okay, so I invest heavily in regulatory teams. In the US, it's FDA. For us, it's Health Canada, looking at coordinating this. It's easy to get a patient into the scanner, into the treatment. It's not so easy getting to that point. Okay, so regulatory is very important. So for that, a high-performance team is really the first, second, and third most important things. The most exciting thing, of course, is that technology is evolving in real time. It's a very different procedure now than it was a few years ago. The first focused ultrasound for tremor case that we did in 2012 took us about 8 hours. Okay, now we can do the procedure in about an hour and a half. Okay, so it's a very different procedure and it continues to get faster. And it's the team that has allowed us at Sunnybrook at least to fire on all cylinders, and very proud to work with this tremendous team that we have at Sunnybrook that allowed us to again fire on all cylinders and lead some of the world first indications in cancer and neurodegenerative disease for both diagnostic and therapeutic indications across the board. So this is the technology today in 2024 that we use with our industry partner Insightec. This is the Exablate magnet, which has a frame, cooled water, degassed in order to allow ultrasound to travel through, all done with a 3T research MR with over 1,000 transducers. We fully anticipate that every aspect of the procedure will evolve in the next year or two. So again, it's evolving in real time. Most procedures are now frameless, pinless. We no longer have to shave patients' head for most indications. Ultimately, we're going to quadruple the number of transducers, which will give us extremely excellent control over microbubble location and activity. And one day, even taking this out of the MRI scanner and doing this in a kind of portable environment. So this very much is the future of non-invasive approaches. So again, the field is rapidly evolving. Where are we going in the next couple of years? We'll expand ablative indications as each of those technical factors begin to be overcome. We're looking at epilepsy, of course, psychiatry, and pain. In the next 5 to 10 years, I think we're going to see much more interest in therapeutic delivery as we work with our partners in biology to try to figure out, well, what is the best therapy to deliver, and ultimately neuroprotective strategies will come in the next decade. So with that, I want to thank the tremendous team I have the great privilege of working with at Sunnybrook and at the University of Toronto. And I'm very lucky to do the work that we do alongside them, and happy to answer any questions. Thanks very much.
Thank you. Great talk. We'll have time for one question.
Yeah, really exciting talk. So as we see this technology evolve, one of the things that would be nice to see is how the blood-brain barrier could be persistently opened. So why is that important? So if we could administer the drug a number of times during that window, which now is very transient, I want to get your thoughts on that. Is that something you see that can happen as this technology gets better and better?
Yes, I mean, the most common question we get is what happens if the BBB is open too long? And, what happens from an immunologic, immunotherapeutic, and other perspective. I agree with you. I think we've done tests and we've done models where we've opened it for much longer. The issue is you need much higher microbubble dosage, and the higher the microbubble, the increased risk of unstable cavitation and bleeding. So it's really a fine balance between how long you keep it open and not. But ideally, the more streamlined the procedure is, the more frequently it could be done.